Hello, Kenzie.
Hi, doctor.
How are you? I heard that you're sick.
Yes, I was very sick last week. That's why I didn't go to school.
I hope you're better now.
Yes, alhamdulillah.
Okay, we'll start exactly in 2-3 minutes.
Okay, will we solve paper 2 and paper 1 or what we are going to do
I
today?
want to finish explaining a part and then we will solve paper one. We have been solving a lot of paper one in school, so we might continue around 10 pages if I finish quickly what I want to finish.
Okay, it's fine.
Okay, exactly 2 minutes at 4 and I'll start. I'll share my screen. Can you see my screen now?
Yes.
Okay, now where are we? Okay, Kenzie, we started in school the lesson protein synthesis. It's actually a very important lesson. I'm very upset that you did not attend. Okay. Now I'll try to give a very, very quick revision over what we took in school, actually. I'll be very quick. This will be revision for everyone, and pay attention, Kenzie, because unfortunately you missed it. We at school, we studied the structure of the nucleotides. The nucleotides, these are the monomers that make up the DNA and the RNA and the ATP. Okay, we have to know the differences between the nucleotides that make up these large molecules. Now, the DNA nucleotide is a macromolecule made up of a pentose sugar and a phosphate group and a nitrogen base. Now, this sugar in the DNA, who will remind me? It is a what?
Deoxyribose.
Deoxyribose sugar. You don't have to know the full structure of the deoxyribose, but you need to know that it is a pentose 5-carbon sugar with carbon number 2 containing a hydrogen atom. This is carbon number 2. Now let's label the carbon atoms so we don't get confused. You have an oxygen here. This is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, and carbon number 5 is outside the ring. Carbon number 2, you always look at carbon number 2, or we call it the 2' position. We have also a nitrogen base. These bases in the DNA are always one of 4 bases. Adenine, bonded always to thymine in complementary base pairing, and cytosine always bonding to guanine. Adenine bonds to thymine with 2 hydrogen bonds, and the cytosine or cytosine bonds with the guanine with 3 hydrogen bonds. So how do we memorize the complementary base pairing? Simply at Cg at 2Cg3. Okay? This is the DNA nucleotide. Now, the RNA nucleotide is very similar to the DNA nucleotide with some differences. Again, we have a pentose sugar, a 5-carbon sugar, but this sugar is a little bit different. What is it? The ribose. Yes, Adham? Speak up with me. The ribose sugar. In the DNA, if you remember, it is the deoxyribose. Also, we have a phosphate group and we also have a nitrogen base, but some things are different. In the pentose sugar, carbon number 2 or the 2' position, we don't have a hydrogen atom, we have a what? A hydrogen molecule. Exactly. This is oxygen 1, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. This is the oxygen position. It's not numbered. We number the carbon only in a clockwise direction. Carbon 1, 2, 3, 4, 5. Carbon number 5 is outside the ring. Carbon number 2, I look at it. I find a hydroxyl group, then this is ribose in the RNA. Now the bases are also different. We have also one of 4 bases, either adenine, but not bound to thymine, bound to what? Uracil. This is a new base that is only found in RNA. It's called the uracil. Okay, adenine always binds to the base uracil, and the cytosine always binds to the base guanine or guanine. Again, they are bound by hydrogen bonds. A binds with uracil with 2 hydrogen bonds, and the cytosine binds with the guanine or the guanine with 3 hydrogen bonds. This is the RNA nucleotide. Any questions? Kenzie?
No.
Okay. So we're just comparing the different nucleotides in terms of molecular structure. And then we studied ATP. ATP is the energy currency of the cell. It's made up of adenosine. The name speaks for itself. Adenosine triphosphate. Tri means 3. So how does it look like? The adenosine is the ribose sugar. Ribose means at carbon number 2 or the 2' position, we have a hydroxyl group. Again, oxygen, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 outside the ring, and the phosphate. And the base is only one base. It doesn't change. It's the adenine base. So the ribose sugar together with the adenine base, this is the adenosine part. And how many phosphate groups do you expect? It's called adenosine triphosphate. So how many phosphate groups
3.
does the ATP have? 3. Okay. Every bond here, it stores energy. And when the bond is broken down, energy is released. And when we add a phosphate, energy is stored. That's it. This is all what you need to know about the ATP. Okay. I'm revising what we took before. Now we also are obliged to know the DNA double helix structure in detail. It is made up of 2 polynucleotide strands. You already know this from before. What is new to you is that these 2 strands are arranged in an anti-parallel direction or position. They are anti-parallel to each other. In other words, one is pointing in one direction and the other is parallel, but it is pointing in the opposite direction. Now, how do we draw that? We will draw one polypeptide chain. If you have a paper in front of you, this will be really helpful. We will draw one polypeptide chain, and we usually choose 3 nucleotides. I will draw the 3 pentose sugars and the bases. And this is a base here. And then a phosphate group protruding outwards like this. Now these nucleotides are bonded together. They're bonded together by this bond here. Now let's look closely at this bond. This bond is made up of 1 phosphate group, as you can see, bonded to 2 carbon atoms at 2 different nucleotides. Let's number the carbon atoms. If this is oxygen, carbon 1, carbon 2, carbon 3, carbon 4, this is carbon 5. And this is also oxygen. This is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So this bond is made up of 1 phosphate molecule, this one, bonded to carbon number 5 in 1 nucleotide and carbon number 3 in another nucleotide. Can you see that? Carbon number 5 in 1 nucleotide and carbon number 3 in the next nucleotide. This bond is very important. It's called who will remind me? I forgot. Who will remind me? What's this bond called, Mariam? Adham?
Phosphodiester bond.
Very good. It's called the phosphodiester bond. Phospho, because it's made up of 1 phosphate molecule. Di, because it is made up of 2 ester bonds. This is 1 ester bond, and this is the other ester bond. Now, why is this bond very important? Because this bond connects the nucleotides together, and it will create the sugar phosphate backbone. This is the sugar phosphate backbone of the DNA molecule. Okay? And this bond is catalyzed by an enzyme. What is the name of the enzyme? Who will remind me? Mariam.
Polymerase.
Very good. The DNA polymerase enzyme. Very nice. By drawing all these molecular structures, you nearly finished half the lesson. Okay, Kenzie? So you need to just draw and remember how to draw these molecular structures. You will be done with what we said. Do you have any questions?
No, not yet.
Okay, not yet. Well, I also unfortunately did not finish because we have one strand looking upwards, just like this one, and we have another parallel strand, but it is looking downwards. Now, here it is a little bit complicated. So I'm going to draw it again for you. I'll try to be as clear as possible. Again, I have 3 sugars with bases pointing sideways. And then at carbon number 5, I have a phosphate group here bonded. So I'm going to project it outwards like this. That is bonded to every nucleotide chain by the phosphodiester bond. This is carbon number 5, and this is carbon number 3. Now, this strand is pointing in one direction. The phosphate is pointing upwards. This strand, we call it in biology the 5' strand. 5' strand. Why did they call it the 5' strand? Because they realized that in this strand, carbon number 5 is the carbon available to bond with the next nucleotide. Let's count. This is the oxygen. Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Carbon number 5 is the carbon that is available to bond with the phosphate here and another nucleotide up there. So that is the carbon available to bond with the next nucleotide strand. So they called it the 5' strand. Now the other strand is looking in the opposite direction. Now, how do we draw that? We did this in class. We'll go to carbon number 1. We will move horizontally as if we will draw an imaginary line, but we will not draw an imaginary line. I'll make a dot here. I'll go to carbon number 1 in the next nucleotide. And again, I'll make a dot just straight in front. I'll go to carbon number 1 and the third nucleotide, and I'll move horizontally and I'll draw a dot. And then I'll go back to my dots. Let me change color. And I'm going to draw an anti-parallel strand that is looking downwards in the opposite direction. How do we do that? I'm going to draw an inverted sugar. An inverted sugar. Now, what was the importance of those dots? So that the sugar is on the same line, because if it's not on the same line, you will not be able to bond the bases together and you will have to erase and repeat, and we don't want to waste time. So I'll show you exactly how you will do it. You will draw an inverted sugar and the base coming out of the sugar. See, the bases now are in one line, so you can connect them together by hydrogen bonds. And these hydrogen bonds, they are represented always by dots. So I have to draw it like this. Dashes or dots. Okay, here is the nitrogen base. See, because we followed the line, so now the bases are in one line. I have a base here that is connected with the other base by 2 or 3 hydrogen bonds. And then let's follow the number of the carbon atoms. This is oxygen, carbon 1, carbon 2, carbon 3, carbon 4. Carbon 5 is outside the ring. Carbon 5 is bonded to the phosphate. So I'll go to carbon 5 and protrude a phosphate. The phosphate is protruded outwards. And I will bond the phosphates to carbon number 3 to create the phosphodiester bond. So did you see, Kenzie and the others, how come I have one strand pointing upwards and another strand that is parallel, but it is anti-parallel? In other words, it is pointing downwards, as if they are, it's not as if they are in opposite directions. Do you see that?
Yes.
Okay. Now you have to know how to draw it and describe it and break it down in detail. It's not difficult. Just practice it 3-4 times and you will realize that it's really, really easy. Okay. Now that other anti-parallel strand, tell me, who knows what is the name of that strand? Because it has a name. The strand with the phosphate looking down.
3' strand.
3' strand. Yes, it's called the 3' strand. And why did they call it the 3' strand, Mariam?
Because the carbon number 3 is available for the next bonding.
Very good. If you look at the 3' strand, you will find that it is looking downwards with the phosphate pointing downwards. This is how you will use to identify it. But they called it, or they gave it that name because they realized that carbon number 3 is the carbon here available to bond with another nucleotide. So they called it the 3' strand. Okay, so we revised everything we took on Wednesday quickly, and it was a good summary. Let's go back to my PowerPoint. This is what we just drew a while ago. We drew the two anti-parallel strands with a 3' end and a 5' end. I hope it's clear. So what are we going to take today? First, we will discuss the frequency of bases, and this is something we took in the O level. They can get you tricky questions and tell you that if 28% of the bases are adenine, how many percentage of bases will be thiamine? Adenine, what is the complementary base to adenine?
Thiamine.
Thiamine. So if adenine is present by 28%, thiamine will also be present by
28%.
28%. Okay, very easy. And then the other two bases, what will be the percentage of each one of them? 28%. So what is the total percentage? 100. I have 28% adenine. I have 28% thiamine. So 28 plus 28 is how much?
56. Both
56.
bases will be 44 in total.
Very good. So the cytosine and the guanine will be in total, how much you said, Adham?
44%.
44. So divide that by 2. Each one will be how much?
22.
Huh?
22.
22. Yes.
20.
They get you yes, you are correct. They get you questions over the frequency of bases, and this is supplement. I mean, this is, yes, O level. Okay, now this is why I met you today, to discuss how the DNA is replicated. And I want you to look at all the GIF pictures here, because this is a very new process here. DNA replication, we know that the DNA replicates before mitosis. In order to produce identical DNA, we produce identical DNA. What happens? I'm going to try to use my diagrams. We have a double-stranded DNA molecule in which the bases are bonded by hydrogen bonds. Several steps happen. Pay attention. Number one, this DNA strand, it will unwind. Unwind means it will uncoil. So it will become a little bit looser, or it will look like this. It will unwind or uncoil. It will not be coiled anymore. It will still be connected. The bases will still be connected, but it will be uncoiled. And then step number 2, the hydrogen bases will disconnect. The hydrogen bond between the bases, the nitrogen bases will disconnect. The hydrogen bond between the bases will be broken down. And this is going to be step 2, and it's going to be called the unzipping. Unzipping. So again, we have a double helix. This double helix is coiled together. The first step that it is going to uncoil or unwind, it's going to look like this. And then that will be step 1. Step 2, it will unzip. Unzip means the hydrogen bonds between the bases will break. This is the unzipping. At the end of the unzipping process, the DNA will look like this. I will have 2 strands with exposed bases. Now, who broke the hydrogen bonds? An enzyme called the DNA helicase enzyme. So are you with me? First, we had a coil of DNA. Number 1, it will uncoil. It will unwind. Uncoiling or unwinding will make it look like this. Step number 2 is the unzipping step. In other words, I'm going to separate these 2 polynucleotide strands. How do I unzip? I will unzip by breaking the hydrogen bonds using the DNA helicase enzyme. So at the end of the unzipping step, I will have two exposed strands with exposed bases. These bases are exposed bases. Are you with me? Are
Yes,
you all
miss.
with me?
Yes.
Okay, very good. Now, what will happen? Step number 3. These exposed bases will attract free activated nucleotides. I have a nucleotide here that will become attracted to the exposed bases. And these bases, these free exposed nucleotides, will bind with the exposed bases by complementary base pairing. Now, where did these free nucleotides come from? They came from a free nucleotide pool in the nucleus. And who will bond the free nucleotides with the exposed bases? The DNA polymerase enzyme. Now, I think my drawings are not very descriptive, so I'm going to use the PowerPoint. So this is step 1. What you see here is step 1. I had a DNA that is coiled. Now, this DNA, number 1, it will unzip and uncoil. It will uncoil and unzip. Uncoil means it will look like this. It was coiled and tightly packed. I'm going to uncoil it and I'm going to unzip. What you're seeing in front of you is the unzipping. What is happening in the unzipping? The DNA helicase enzyme is breaking the hydrogen bonds between the bases. So we will end up with two strands, the two polynucleotide strands with exposed bases. Are you with me? Are you with me?
Yes.
Good. So this
Yes.
is the first step. This first step, we usually write it together. It is called unzipping and unwinding. But if you want to be very precise, the unwinding happens first, or the uncoiling, followed by the unzipping. The unzipping means I'm going to break the hydrogen bonds between the bases so that I have two exposed strands. And who will break the hydrogen bonds? The enzyme?
Helicase.
Helicase, the DNA helicase enzyme, it's like the scissors. It will break the hydrogen bonds between the bases in order to expose the two polynucleotide strands. Now, every exposed polynucleotide strand, this is one of them, will be used to create the new complementary strand. So every old exposed polynucleotide strand is called the template. This word is very important. What will happen next? So here, this is exactly what we're doing. This is the unzipping together with the creation of the new complementary strand. Let me show you another picture. Okay, here I have, it should be moving now. Yes, here I have an exposed strand that will act as a template. What is arriving here? A free activated nucleotide with bases that will bond with the exposed bases by the rule of complementary base pairing. A binds with T and C binds with G. Who will help in the bonding of the bases and the making of the phosphodiester bond? The DNA helicase enzyme. So let's look at the pictures. Let's take it step by step. By the way, it's very easy. Step number 1, I'm going to uncoil. I had a coiled DNA molecule. I will uncoil it so that it looks like this. Step number 2, I will break the hydrogen bonds between the bases, so I separate the strands. That step is called unzipping. Who will break the hydrogen bonds between the bases? The DNA helicase enzyme. At the end of that step, I'm going to have two exposed strands. These strands will act as the template, or they will help me to create the new complementary strand. So they will act as something called the template. Are you all with me?
Yes. Yes.
Very good. So now I have two exposed templates. These exposed templates with exposed bases, those bases will attract free activated nucleotides. Where do these free activated nucleotides come from? They're found in the nucleus. They come from a nucleotide pool in the nucleus. Okay? These free activated nucleotides, as you can see, they will bind to the exposed bases by complementary base pairing, and the hydrogen bonds will be made. And every new nucleotide will bind with the previous nucleotide by the phosphodiester bonds. And who's going to do all that? Which enzyme will create the new complementary strand?
DNA polymerase.
DNA polymerase. Very good. Now I want to stop here a little bit. I want you to look at the free activated nucleotide. This free activated nucleotide is a little bit different from the normal nucleotide that we studied. The normal nucleotide that we studied looks like this. We have a deoxyribose sugar, and we have a nitrogen base, and we have only one phosphate. Now I want you to look at this free activated nucleotide. Let's look from the beginning of the picture. Okay, look at this free activated nucleotide. It's going to arrive now. How many phosphate groups does it have?
3.
1 or 3? 3, exactly. So the free activated nucleotides, this one, it keeps on moving. It has 3 phosphate groups. It has 2 additional phosphate groups. These 2 additional phosphate groups are the groups that activated the nucleotide. And who is going to break off these 2 phosphate groups? The DNA polymerase enzyme. When these 2 phosphate groups are broken, they release energy. And this is the energy that is needed to create the phosphodiester bond. So I'll break it down again so it's nice and easy. Okay. Everything at the beginning looks difficult, and then you will realize that it's really actually easy and interesting. So what are we studying? We are studying DNA replication. How do we have an initial DNA molecule that is a double helix, and we end with 2 identical DNA molecules? They are identical. Now what happens? The DNA is coiled. Step number 1 is uncoiling and unzipping. Uncoiling, let's take it step by step. Uncoiling means the DNA will look like this. It will uncoil. Okay, this is easy. And step 2, the hydrogen bonds will break so that we separate the 2 polynucleotide strands. That step is called unzipping. Who will break the hydrogen bonds? An enzyme called the what? The scissors.
DNA helicase.
Very good. The DNA helicase. So after we break the hydrogen bonds, we will have 2 templates. What are the templates? Templates are the original DNA strands that will help us create the new complementary strand. This is the definition of a template. A template is the original. You have to use the word original. It is the original DNA strand that we will use to create the new complementary strand. Very good. Now we have 2 exposed templates, or templates with exposed bases. Who will arrive and bond with the exposed bases? These exposed bases, what do they attract? Free
Nucleotide bases.
Very good. Please use this term, free activated nucleotides. Where do they come from? They're found in the nucleus. And why are they activated? Because we have 2 extra phosphate groups bound to them. This is the normal nucleotide, a deoxyribose sugar and a base and 1 phosphate. They have 2 extra phosphates. These 2 extra phosphates make them activated. The DNA polymerase enzyme will break these 2 phosphate groups. Breaking of these phosphate groups releases energy. The DNA polymerase enzyme will use this energy to bond the new nucleotides together, creating the phosphodiester bond and the sugar phosphate backbone of the new DNA molecule. What do you think? So let us just take it step by step. I'm not finished. Let's take it step by step. This method of semi-conservative replication, or this method of replication, is known as the semi-conservative DNA replication. And we will understand why they called it the semi-conservative replication. Let's take it step by step. Step one, we will separate the DNA molecule. How does this happen? By the unwinding and the uncoiling and the unzipping. In the unzipping, we have to describe unzipping by saying that we will break the hydrogen bonds between the bases with the help of the DNA helicase enzyme. And this will result in 2 template strands with exposed bases. Easy? I have a DNA that is coiled. It will unwind and it will unzip. Actually, you can explain all the steps with drawing. This is the starting molecule. I will unwind, it will look like this. I will unzip, it will look like this. Unzip means I will break the hydrogen bonds and separate the bases. And now I have 2 templates with exposed bases. Until this point, everything is clear? Hard? No, good. And then who's going to arrive? This is the unzipping. And these are the free activated nucleotides that bind by complementary base pairing with the help of the DNA polymerase. Again, here I have an exposed template, a template with exposed bases. The free activated nucleotides will arrive. They are activated because they have 2 extra phosphate groups. These phosphate groups, the 2 extra ones, will be broken by the DNA polymerase, and this generates energy. And this energy is going to be used to bond the new nucleotides together, creating the phosphodiester bond and the sugar phosphate backbone. Nice? Let's read it together. So, the replication step, or the replication phase, we start with the word free activated nucleotides. They will arrive from a nucleotide pool in the nucleus. They will bind with the complementary bases on the exposed strands. And who will help? The DNA polymerase enzyme. How does the DNA polymerase enzyme do that? It breaks off the 2 extra phosphate groups on the activated nucleotides. This releases energy, and this energy is going to be used to create the 1 phosphodiester bonds between the nucleotides of the new strand. And the making of these bonds releases water, and it's a condensation reaction. 2, creating the sugar phosphate backbone of the new strand. What do you think? Anyone has any questions? Kenzie?
No.
Okay.
Yes, doctor.
Okay. Kenzie, you have any questions?
No.
Now, I will mention 1 point in this replication process, and I will be done. The DNA polymerase enzyme works in a specific method. It has a specific way of operation. This picture is supposed to move. Why is it not moving? Yes. Okay. The DNA polymerase enzyme, this nice enzyme here, it only binds to the 3' end of the DNA. It only binds, pay attention to this sentence. The DNA polymerase enzyme only binds to the 3' end. Don't forget that the DNA is made up of 2 polynucleotide strands, and they are antiparallel. In other words, one strand is a 5' strand and the other strand is a 3' strand. When the strands separate with the help of the DNA helicase enzyme, the DNA polymerase will not be able to bind with both strands. It will be able to bind to one of them, the 3' strand. So pay attention to this sentence. The DNA polymerase enzyme binds to the 3' strand and builds the new complementary strand from the 5' to the 3' direction. Are you with me?
Yes, miss.
Okay. When the DNA is separated, I have 2 strands that are antiparallel. They are not in the same direction. One strand is a 5' strand with the phosphate pointing upwards, and the other strand is the 3' strand with the phosphate pointing downwards. The DNA polymerase will bind with which one of them? It will bind with the 3' strand. Therefore, building the new complementary strand in an opposite direction, So the DNA polymerase always binds to the 3' strand to build the new strand from the 5' to the 3' direction. This strand that it will bind to, the 3' strand that it will bind to, will create the complementary strand continuously in one go. That strand that it will create will be called the leading strand. Let me look at another picture. Okay, now I want you to focus on this template strand. Ignore this one now. This is the DNA helicase. The DNA helicase enzyme is breaking the hydrogen bonds and it is unzipping. It is exposing the DNA strands to give me two templates. This is one template and this is another template. What is the template? The template is an original DNA strand that will be used to create a new complementary strand. These strands are anti-parallel. They move in opposite directions. One strand is a 3' strand, and the other strand is a 5' strand. The DNA polymerase cannot bind to both strands at the same time. It will bind to one of them. Which strand will the DNA polymerase bind to? The 3' or the 5'?
3'.
Very good. The DNA polymerase, this is the DNA polymerase. It will bind to the 3' strand and build the new complementary strand in an anti-parallel direction. So it will bind to the 3' strand to build the new strand from 5' to 3'. This strand is from 3' to 5'. It will bind to it and create the opposite complementary strand, which is 5' to 3'. Are you all with me? Because the opposite strand or the other strand is anti-parallel. It has to be pointing in the opposite direction. Are you all with me?
Yes, miss.
There's no problems in this strand. So this strand will be produced in one go, continuously. That strand is going to be called the leading strand that is built continuously. Now, I'm sure you don't understand why do they give it this name. You will understand why when we look at the second strand. I want you to look at the second strand. This second strand is the 5' strand. The DNA polymerase cannot bind to it just then. What happens is that the DNA polymerase will wait. Will wait for what? Will wait until a certain length of this strand has been exposed so that the DNA polymerase goes backwards, as you can see, to bind with the 3' end and then moves forward. And then by the time the DNA is synthesizing this section, it will move backwards because another part will be exposed. It will bind to the 3' end to build the new strand from the 5' to 3' direction. And then by the time it is building this section, the DNA helicase would have done some zipping. It will go unzipping and then it will go backwards and move forwards. It will go backwards and move forwards. It will go backwards and move forwards. So that strand is going to be produced in sections. And that strand is not going to be produced continuously. That is why that strand is going to be called the what? Did you hear about it, Adham? The lagging
strand.
Very good. The lagging strand. The strand that is going to be produced in sections. These sections, they have a name, Adham. What is their name?
Okazaki fragments.
Very good. Okazaki fragments. And who will join these fragments together?
DNA ligase.
DNA ligase. Very good. Okay. So let's look at this diagram. Maybe it will make things clear. I have a DNA molecule. This DNA molecule, I want to replicate it. So it will have to uncoil and unzip. It is unzipping here. Who will help in the unzipping? Which enzyme?
DNA helicase.
DNA helicase. Very good. How does it do the unzipping? Which bonds does it break? The hydrogen bonds between the bases. Okay? So the DNA helicase is moving in this direction. As it is moving in this direction, it is breaking the hydrogen bonds. This point of replication has a name, Adham. What is it called? Replication. Replication what? Replication fork. Okay? Good. Now, when we expose the strands, these strands are anti-parallel. I will have a 3' strand and another anti-parallel strand that is 5'. The DNA cannot bond with this one. This is how the DNA operates. It cannot bond with the 5' strand. So it will bond with which strand? Yes, Kenzie, the DNA polymerase will bond with the 3' strand or the 5' strand?
The 3' strand.
Very good. The DNA polymerase will attach to the 3' strand of the original DNA, and it will build a complementary strand. This strand that is complementary will be anti-parallel. So the DNA polymerase attaches to the 3' end of the original DNA strand, building the new strand in the opposite direction, building the new strand in the 5' to 3' direction. So I am the DNA polymerase. I will bind to the 3' end, and I will build a new complementary strand that is anti-parallel. And I'm going to build this in one go, in a continuous pattern. So that strand will be called the leading strand. Nice? Easy? I'm going to bond the new nucleotides together and create the phosphodiester bond and the sugar phosphate backbone. I will have no problems at all. Now, what about the other strand? I cannot bond to this strand because this is the 5' strand. I have to wait a little bit until a section is exposed so that the DNA polymerase goes backwards and binds to the 3' end. This is the 5' end and this is the 3' end. It will go bond here and then move forward and build a section. And then while it is building this section, the DNA helicase would have exposed another part of the DNA. The DNA polymerase will move backwards from here to bind to the 3' end and then move forward to create this section. And then by the time it's creating this section, another part will be exposed. The DNA polymerase will move backwards and bind with the 3' end to build the new strand from the 5' to 3' direction. And this strand will be produced, yes, but it will be slower. It will be produced in sections called Okazaki fragments. And that strand, because it is going to be produced after the leading strand, they called it the lagging strand. Why the lagging strand? Because it's going to lag behind. It's going to be produced in fragments, in sections. And the names of those sections, they take after the name of the scientist. He was called Dr. Okazaki. So they called these sections Okazaki. After these sections are made, they will fuse together with the help of the DNA ligase enzyme. The DNA ligase enzyme, we took this name in O-level, will create the phosphodiester bonds and the sugar phosphate backbone of the sections. I hope this is better. Let me see a new picture. Okay, this is the picture. You have this in your notes. Here is the unwinding and the uncoiling. The unwinding and the uncoiling is usually written as the first step, step one. Step one in the DNA replication is unwinding or uncoiling. What happens in the unwinding and the uncoiling? I have a DNA helicase enzyme that is breaking the hydrogen bonds. Okay? And the point of replication is known as the replication fork. Are you all with me?
Yes.
Very
Yes,
good.
yes.
As I am breaking the hydrogen bonds, I will expose template strands. I will expose the 2 polynucleotide strands of the original DNA that are now called template. But one of the strands is a 3' strand, and the other strand is an anti-parallel 5' strand. Now, the DNA polymerase will only bind to the 3' strand for now. It will attach itself to the 3' strand to build the new complementary strand from the 5' to the 3' direction. It will build the new complementary strand in the opposite or anti-parallel direction. And it's going to build this new strand in one continuous go, not in sections. So this strand is going to be called the leading strand or the continuous strand. Now let's read together. The DNA polymerase, don't know what is happening, but we will continue. The DNA polymerase can only add nucleotides in the 5' to the 3' direction. Now, this is always a point of confusion, how it is said. So I have it here for you clearly. The DNA polymerase attaches to the 3' end of the original DNA, building the new strand. Always write it like this because this is very clear. It will bind to the 3' and build the new strand from the 5' to the 3' direction. Okay? So the DNA polymerase, this is the DNA polymerase. It will bind to this one and build this one. Are you all with me?
Yes.
Okay? Very
Yes,
good.
miss.
Now, what about the other strand, the 5' strand? The DNA polymerase cannot bind to it just yet. It will wait a little bit until part of it is exposed. The DNA polymerase will move backwards. This is 5' and this is 3'. So it will attach to the 3' end, the exposed 3' end, to build the new strand from the 5' to the 3' direction. Look at the colors. The blue strand is the strand that it binds to, and the red strand is the new strand that is made. The blue strand, it cannot bind to the 5' end. It will have to bind, it will have to go towards the inside. It will move backwards, like it will move like this, and then move forward. Move backwards, and then move forwards. Move backwards, and move forwards. So this strand will be produced in sections called Okazaki fragments. That strand will lag behind, and it will be called the what? Lagging strand. What do you think? Okay, this is a very nice picture. Here, I have two exposed templates. This is one template, the blue, and this is another template. One template is a 3' strand, and another template is a 5' strand. The DNA ligase will bind with which strand? The 3' or the 5', Kenzie?
The 3'.
Very good. When it binds to the 3', it will build the new complementary strand in an anti-parallel direction. So this is exactly how we write it. The DNA polymerase attaches to the 3' end to build the new strand in the anti-parallel direction, or to build the new strand from the 5' to the 3' direction. It is attaching to the blue strand, and it is building the red strand. It attaches to the 3' strand to build the new strand from the 5' to the 3' direction. In this case, this strand will be called the leading strand because the synthesis of this strand is continuous in one go. Now the other strand is a 5' strand. I cannot bind to it. I have to wait until a section is exposed to go to a 3' end. This is 5', 3'. Okay? So the DNA polymerase will move backwards and bind to the 3' end and then move forward. And then it will go backwards and then move forwards. And then go backwards and move forwards. And go backwards and move forwards because it has to bind to a 3' end. This strand is going to be built in sections. So it is going to be called the lagging strand. And these sections are called Okazaki fragments. After I finish synthesizing those fragments, I have an enzyme called the DNA ligase that will fuse the fragments together. And I will be finished. Let's read along. The lagging strand, the synthesis of this strand is not continuous. The lagging is created in short segments, which are then joined by the DNA ligase. Let's read this. The DNA ligase joins up the segments of the lagging strand by catalyzing the formation of the phosphodiester bonds between the sugar and the phosphate groups of the segments. Okay, now I want to summarize all this because see this question, I put it for you here. This question is very common. Discuss DNA replication. Outline the role of the DNA polymerase in the replication of DNA. Actually, here they want only the function or the role of the enzyme, but in other questions, they ask you discuss the replication. 5-6 marks. Now, what do? We want the 5-6 marks. We can, number one, draw a diagram, and this diagram will really help us. We can draw the diagram like this. If you have a paper, You can follow with me. I will start with the DNA molecule, which is a coiled double helix. Step number 1, it will uncoil. Step number 2, it will what? What is happening? This is uncoiling. What is happening, Kenzie? I'm breaking the hydrogen bonds to expose the templates.
Uncoiling.
This was uncoiling. Number 1, we uncoil. Number 2, when we break the hydrogen bonds, this is what? Unzipping.
Unzipping.
As if I have a zip and I will unzip it. Okay? This unzipping, I will describe it. I will break. I'll write it with you word by word. I will break the hydrogen bonds between the what, Mariam? Between the bases, okay, to expose 2 original strands, 2 original polynucleotide strands or 2 original DNA strands, whatever. And each strand will now be used to create a new complementary strand. Each strand is called the what? What's the keyword?
Template strand.
Template strand. What is the name of the enzyme that broke the bonds? Helicase enzyme.
DNA helicase.
Good girl. Okay. Now I have 2 exposed strands, but they're not moving in the same direction. One strand is a 3' to 5' strand, and the other strand is a 5' to a 3' strand. Now tell me, Adham, the DNA polymerase will bind with which one?
DNA polymerase 3 prime.
Very good.
قصدي 3 prime.
Yes, it will bind with the 3'. It will bind with both, but it will just build one strand faster than the other. So the DNA polymerase will bind with the 3' strand to build the new strand in the 5' to the 3' direction. Yes? Exactly, This is how we write it. The DNA polymerase, it will bind to the 3' strand to build the new strand in which direction? In the, if it binds to the 3', then it will build the new strand in the anti-parallel direction, which is in which direction? Will it build it also in the 3' to 5' direction, or it will build it from the 5' to the 3' direction?
5 prime to 3 prime.
Exactly. So it will attach or bind to the 3' end to build the new complementary anti-parallel strand from the 5' to the 3' direction. Now, this strand will be called the leading or the lagging strand. It is going to be produced or built continuously in one go. So this is the leading strand. Very good. Now the opposite strand is a 5' strand. Can the DNA polymerase bind to this prime end? Tell me, Kenzie, can the DNA polymerase bind to this prime end?
No.
No, it will have to move backwards or move anti-parallel to bind to the 3' end and then move forwards. So it will build a section. By the time it builds this section, another part of the DNA will be exposed. The DNA polymerase will move backwards to bind to the 3' end and build the new strand in the anti-parallel direction. By the time it builds this section, another part will be exposed, and so on. So that strand is going to be called the leading or the lagging strand. The strand that is built in sections is called the leading strand or the lagging strand. Lagging strand. Are you with me, girls?
Yes.
Good. And the sections are called Okazaki. And after we're done, these sections will be joined by phosphodiester bonds by or with the help of the DNA ligase. And I'll give you a very, very easy tip to identify the leading and the lagging strands. You have a picture. Let me use this one. You have a diagram on the exam, and they want you to label the leading strand and the lagging strand, and they did not tell you the templates are 3' or 5'. Then what do? You will go look at the DNA polymerase enzyme. If the DNA polymerase enzyme is moving towards the replication fork, then this strand it is building is the leading strand. If the DNA polymerase is moving away from the replication fork, then this strand it is building is the lagging strand. I'm going to erase everything I have on this slide, and I want you to really focus on the action of this DNA polymerase. This one. Look, it goes backwards and then forwards. And then it will finish this section. It will go backwards. I don't know why it is slow. It will go backwards. It's not moving. It will go backwards. It will bind to the 3' end and then move forward. It will make a section, and then it will go backwards, and then it will move forward. This strand is built in sections, and this will be the lagging strand. How do I identify it? I will see the DNA polymerase moving away from the replication fork. What do you think? Okay. At the end of the DNA replication process, I will have 2 new DNA molecules. Every molecule will be made up from 1 original strand and 1 new complementary strand. That is why they call this method the semi-conservative DNA replication. Why did they call it the semi-conservative? Because we conserved 1 original template strand and we used it to create the new complementary strand. What do you think? Okay, it might look confusing, but trust me, you will find it very, very easy with time. Let's solve this question, because this question asks about every single detail. Mariam, helicase enzyme allows the DNA double helix to unwind and the hydrogen bonds between the 2 strands to break, exposing the 4 bases. You are expected to give the examiner the name of those bases. Very easy. What is A?
Adenine.
Okay. What is T?
Thymine.
Thymine. What is C?
Cytosine.
Okay. What is G?
Guanine.
Guanine. Easy. Now, this is not what I wanted, actually. And then, yes, an enzyme molecule attaches to each of the 2. No. It's not the right time. Okay, an enzyme molecule attaches to each of the 2 separated parenteral strands. The 2 enzyme molecules move in opposite directions, each catalyzing the formation of a new strand of DNA. This enzyme is known as what? What is the enzyme that is building the new complementary strands, Adham?
DNA polymerase.
Good boy. So I'll write here DNA polymerase. Okay, and why is it moving in opposite directions? Because it binds to the 3' and will build the new strand in 1 continuous go, and it will always have to go backwards and forwards to bind to the 3' end. Okay, Kenzie, DNA, the monomers of DNA are activated with 2 additional phosphates and are free in the nucleus for the synthesis of new strands. What are the monomers of the DNA? What are they called?
DNA is a protein, right?
No, DNA is not a protein at all. DNA is a polymer made up of small monomers. What are the monomers that make up the DNA?
The nucleotides.
Yes, the DNA nucleotides or the monomers of DNA, they are activated. Look at this. With 2 additional phosphates, the DNA nucleotide is made up of 1 deoxyribose sugar and a nitrogen base and 1 phosphate. When we have 2 additional phosphates, they will become free activated nucleotides. They are activated due to the presence of the phosphates. Now they're called phosphorylated nucleotides. And the DNA polymerase will break those phosphate molecules, releasing energy. And this energy is going to be used in the building of the DNA molecule. Let's look at number 4, Mariam. The bases of the DNA monomers form hydrogen bonds with the bases on each separated parenteral strand of DNA, according to the rules of, now this is easy, This is O level. When adenine binds with thymine and cytosine binds with guanine, this is called complementary what?
Base pairing rule.
Yes, complementary base pairing. Okay, Adham. One DNA strand is synthesized continuously, and the other is synthesized in sections, known as the Okazaki fragments. The fragments are joined by an enzyme. Which enzyme?
DNA polymerase enzyme.
No, the DNA polymerase
Ligase,
enzyme.
ligase.
Yes, the DNA ligase enzyme. Okay, which catalyzes the formation of the phosphodiester bonds. Okay. The result of the replication is two DNA molecules, each one containing an original parental strand and a newly synthesized strand. This type of replication is described as what, Maryam?
Semi-conservative.
Good girl. Semi-conservative DNA replication. Good. Okay, I'm not going to explain anything more. Don't worry, because this is exactly what I wanted to explain today, the DNA replication. I will use the pictures to help. This is the original DNA strand. When the hydrogen bonds are breaking between the bases, Kenzie, what step is that?
Unzipping.
Very good, unzipping, where the hydrogen bonds are breaking. What is the name of the enzyme Adham that breaks the hydrogen bonds?
DNA polymerase enzyme.
Helicase, good boy. So this is step one. I will unwind and unzip and explain what unzipping is. I will break the hydrogen bonds. Now, who will arrive from a free nucleotide pool to bind with the exposed bases, Maryam? What are these, my dear?
Free activated nucleotide.
Good girl. Okay, they're very obvious. I can see two extra phosphates. So these are the free activated nucleotides. And how did I know, Maryam, that they are activated? What do they have extra? Okay, your mic is not very efficient, but I think you said two extra phosphates. These two extra phosphates will make them activated. They have another name, they're called triphosphatate, but the free activated nucleotide is an easier name. Okay, Adham, who will break these two phosphate molecules to generate energy? Which enzyme?
DNA polymerase enzyme.
Excellent. The DNA polymerase enzyme will break these two phosphates to generate energy, and this energy will be used to generate the phosphodiester bond and the sugar phosphate bond. Look at this. These are the activated nucleotides. How did I know? Because I can see the nucleotide with two phosphate groups. These phosphate groups will be released. They will be broken down. Who will break them down? Just like Adham said, the DNA polymerase. As they are released, this will give us energy. And this energy will be used to bind the new nucleotides together, generate the phosphodiester bond, and the sugar phosphate backbone of the new strand. This is the original strand. As long as this is the original strand, then we call it the template strand from the original, okay? And this is another template strand because I'm going to use both template strands to make or to build my new strands. Let's read together. I'm not going to explain anything new. I'm just going over what we said. DNA polymerase catalyzes the formation of new phosphodiester bonds to create the sugar phosphate backbone of the two DNA strands. What do you think? Anything is wrong? Anything is hard? If I want to summarize that, I will say the DNA replication. The DNA will unwind. 2, the DNA will unzip, or you can put them both in one step. And you can explain what is unzipping. The hydrogen bond will break with the help of the DNA helicase. Step number 3, you will have to mention that now you have two original strands with exposed bases, and these are called templates. And they will be used to build the new complementary strand. And here, that part, you have to mention that one template will be 3' and the other template will be what? Anti-parallel to it.
5'.
If one template is 3', yes, the other template will be 5'. Here you have to start talking a little bit advanced and mention that the DNA polymerase will bind to the 3' to build the new strand in the anti-parallel direction. It will bind to the 3' end to build the new strand from the 5' to the 3' direction. If it is building the strand continuously and moving towards the replication fork, this will be the leading or the lagging strand, Kenzie?
The leading strand.
Leading, because I'm going to build it continuously. If the DNA polymerase will go backwards and then forwards and backwards and then forwards to build this strand in sections, this will be the leading or the lagging strand, Mariam?
The lagging strand.
Yes. Okay. Now this picture, does it show us anything? It doesn't show us anything. I don't know why did I use this picture, but okay. Now these are the steps written in your notes. I will not read them with you. It will be too boring. Okay. I hope you understand. This is the semi-conservative replication. We will use 2 original strands to act as a template, and every original strand will be available or present in the final DNA molecule. That's why we will conserve one of the strands in every DNA molecule. That's why it is called the semi-conservative replication of DNA. Okay, I will stop here. I promised you that I'm only going to explain this part. This part is very important. It's very easy, and it's only easy if you understand it. Don't memorize anything. Let me see what this picture, okay, these are the enzymes involved. The helicase, we did not take the primase, DNA polymerase to make the phosphodiester bond and the sugar ligase, and the ligase, the phosphodiester bond and the sugar phosphate backbone, the ligase for the fusion of the Okazaki fragments. Now this picture is nice because this picture clearly shows you that the DNA polymerase in one of the strands, it is moving continuously. And in the other strand, it goes backwards and then forwards, and then backwards and then forwards, and then backwards and then forwards. That will be the lagging strand. Okay, I promised I will only explain this part. Anyone has any questions?
No, miss.
No.
Is everything clear?
Yes.
Also, this
Yes.
picture is nice. I'll send you a video, by the way. I'll look for a nice, clear video, and I'll send you a video. Maybe the video will have more information. You only need to know what I told you. Here, the DNA polymerase, it will bind to the 3' end to build the new strand from the 5' to the 3' direction. It is moving towards the replication fork, and it is building the new strand continuously. This will be the leading strand. Now, in the opposite strand, it cannot bind to this 5' end. It will have to move backwards to bind to the 3' end, and then move forwards. And then move backwards to bind to the exposed 3' end, and then move forward. And then move backwards, and then move forwards. So this strand will be built in sections called Okazaki fragments, and that strand will be called the lagging strand, and these fragments will then be joined together by the DNA ligase that will join the phosphodiester bonds of the segments and the sugar phosphate backbone of the segments. You are not requested to know what the primer does, but in case you're asked, this primer indicates the starting position for the DNA polymerase. That's it. And the helicase enzyme is here. It is cutting or breaking the hydrogen bonds, and the point of replication is called the replication fork. Okay, I'm done. Anyone has any questions? This is only a section. It's part of the lesson. Please read it. I just want you to read it. I don't want you to study it. I want you to read it and make sure that you understand it. If there is a word or a sentence that you don't understand, please ask me. Can you do this for me today?
Yes, miss.
Okay. Thank you so much. Okay. Have a good day. Bye.
Bye.